The overall goal of this procedure is to examine the ability of a probiotic Streptococcus salivarius to inhibit pneumococcal adherence to an epithelial cell line. This is accomplished by first preparing CCL23 epithelial cell monolayers in a 24-well plate. Next, S. salivarius and pneumococci are added to the monolayer in sequence, and the cells are incubated. In the final step, the wells are washed to remove the non-adherent bacteria, the CCL23 epithelial cells are lysed, and the well contents are harvested. Ultimately, the impact of S. salivarius on pneumococcal adherence can be evaluated by quantifying the number of cell-associated pneumococci.
This method helps answer key questions in the infectious diseases field, such as whether probiotics can inhibit the adherence of bacterial pathogens such as the pneumococcus.
We first had the idea for this method when we began investigating the potential use of Lactobacillus rhamnosus GG and other probiotics in the respiratory tract.
Demonstrating this procedure will be Jayne Manning and Zheng PhD students from our laboratory.
On the day before the experiment, split a culture of confluent CCL23 epithelial cells and then dilute them in pre-warmed media to a 150,000 cells per milliliter concentration in a sterile 50 milliliter tube. Next, seed 1 milliliter of the cells per well in a treated 24-well tissue culture plate and incubate the cells overnight. Then, using a 10 microliter loop, inoculate 10 to 12 well-separated colonies of each bacterial culture into a 10 milliliter aliquot of Todd Hewitt broth, plus